Hi students, and welcome to your first lecture in gene regulation in eukaryotes. I'm Dr. Jeevan, and I will be lecturing you for the first part of the course. In this lecture, we will begin with Section A, Structure of DNA and Epigenetics. The learning objectives that we will complete in this lecture are to describe the structure of chromatin and explain how dynamic movement of chromosome compartments facilitate gene regulation, and we'll also be describing the structure of a gene briefly. Expansion into these other concepts will be continued in lectures that are to follow. By now, we should all be familiar with the central dogma of molecular biology. The central dogma states that genes that are contained with the DNA hold the sequence or the code for a template strand that can be transcribed into mRNA. The mRNA contains the triplet code words that are required to convert that mRNA into a functional protein. Mutations at the DNA level or at the RNA level can result in insertion or deletion mutations or point mutations, which can alter this triplet code and lead to a mutated protein product. Of course, a mutated protein product may be non-functional or have a partial function and therefore may not serve to play the role that it was originally intended if it's mutated. Therefore, tight regulation of transcription and translation is important for proper gene function. Studies on protein and RNA content have shown that the amount and the type of protein or RNA molecules that are expressed in the cell can differ between different cell types. However, DNA content is stable across all cell types within an organism. Multicellular eukaryotes are composed of different cell types. For example, we have liver cells or skin cells or cells that have different levels of differentiation. And so if the DNA content is stable across these cell types, then that variation or the variable differentiation states must be attributed to the differences in protein and RNA content in these different cell types. And that's what we plan to study in this course. Cell differentiation can be regulated by variations in gene expression patterns because the genes that are expressed will determine which mRNAs are present and which proteins are present in that cell type. Genes can be expressed in a process called transcription, and this process requires an activator. An activator can be an extracellular protein that triggers an intracellular signaling cascade, or cascade of events that eventually leads to the expression of specific genes and specific proteins. So if we look at this figure on the right, you will see two different cell types, oviduct cells and liver cells. And they've both been exposed to an activator called estrogen, which is a hormone in this case. In oviduct cells, estrogen leads to the transcription of the ovalbumin gene, but it does not promote transcription of the vitellogenin gene. Whereas in liver cells, exposure of these cells to estrogen will lead to transcription of the vitellogenin gene, but not the ovalbumin gene. And so what this tells us is that transcriptional activation alone is not sufficient for adequate gene regulation. As you can see, exposure to an activator alone can lead to the expression of two different genes completely in different cell types. So we need to understand that gene regulation is a lot more complicated than simply expressing a particular mRNA, and this process is tightly regulated at various levels. Now going back to the central dogma. This diagram shows the typical process of transcription and translation in a eukaryotic cell. So we'll begin with chromatin. Chromatin first has to be remodeled in order to expose the correct regions of DNA that are to be transcribed. The DNA is then transcribed into a pre-mRNA sequence, which is further processed through splicing, capping, and poly-A tailing. Once the mRNA is exported into the cytoplasm, it should then be stabilized and occupy a specific location before it is translated into a protein product. This entire process is tightly regulated at various levels. We'll be discussing transcriptional regulation and post-transcriptional regulation, and then in the final week, we'll be talking about translation. So when we talk about transcriptional regulation, we refer to how the process of transcription is controlled. And transcription can be regulated at various levels. Firstly, genome organization. Because the chromatin has to be remodeled to expose specific regions of DNA, the region of chromatin that is exposed in order to be transcribed serves as the first point of regulation. So we'll be discussing eukaryotic genome organization. We will then talk about how chromatin can be remodeled. So how are certain regions exposed and other regions not exposed at different points in the cell cycle or in different cell types for different levels of differentiation. After discussing chromatin remodeling, we will then discuss how DNA and histones can be epigenetically modified. So epigenetic modification involves addition or removal of certain functional groups in order to regulate how DNA is exposed. We will then talk about post-transcriptional regulation, and that is how mRNA is regulated after it's transcribed. And post-transcriptional regulation refers to mRNA processing and nuclear export, as well as mRNA stability and localization. Before we continue with discussing chromatin structure and genome organization, it's important for us to understand the concept of a gene. So early studies conducted back in the 1950s by scientists like Gregor Mendel, as well as early gene mapping studies on Drosophila, led to the first definition of the gene as a hereditary unit organized in a specific location along a chromosome. Once Watson and Crick defined the structure of DNA, the gene was then defined as a sequence of nucleotides in a DNA helix that encodes genetic information. In the 1960s, after mRNA splicing was discovered and the discovery of introns and exons, this led to reverting back to an old understanding or definition of the gene. And that understanding was one gene equals to one enzyme, or the one gene to one enzyme hypothesis. And that hypothesis suggested that a single gene would encode a single polypeptide or a single mRNA. The definition was further expanded to include non-coding regulatory sequences upstream of the coding region. So this definition of the gene, or the concept of the gene, is what's largely accepted and used as a definition now today. However, more recently, we've discovered non-coding RNAs that are also transcribed but not translated into proteins. And this has led to a new debate on adjusting the concept and the definition of a gene. For now, we will consider a gene as it's described here. A eukaryotic gene is composed of a promoter, which contains upstream regulatory elements, as well as the transcribed region. Now, these upstream regulatory elements are not transcribed. However, they play an important role in regulating how a gene is expressed. And so the promoter region as well as the transcribed region of a gene are both considered to be part of the gene. The transcribed region contains both 5' and 3' untranslated regions. Both exons and introns are transcribed to form a pre-mRNA, which are later spliced in order to form the preliminary mature RNA. After that spliced mRNA is capped and tailed, it is then referred to as a mature RNA. So we need to understand that a gene is not just the coding region or the coding sequence anymore. In eukaryotes, when we talk about a gene, we refer to both the upstream regulatory elements or the transcription factor binding sites. These regulatory elements can also include binding sites for repressors, enhancers, or silencers. And the entire transcribed region is also not translated into the protein. And therefore, all these regulatory elements play an important role in regulating how a gene is expressed and when a gene is expressed. So we need to understand that when we're thinking about a gene, it's not just the coding region. It's the transcribed region as well as the regulatory elements that are involved in determining when that gene is expressed. We'll now talk about genome packaging in eukaryotes. So the primary structure of chromatin is double-stranded DNA. And that double-stranded DNA contains our genes. Double-stranded DNA is first wrapped around an octamer of histones to form the nucleosome. The nucleosomes are then further coiled to form a 30 nanometer chromatin fiber. And these fibers form 300 nanometer loops, which are further compressed to form the chromatid and a chromosome, which has a total width of 1400 nanometers. Chromatin is most firmly compacted during metaphase. However, during the rest of the cell cycle, chromatin is more loosely packed to allow for gene expression. This diagram will help you understand what was described in the previous slide. We'll start off with the DNA double helix, which is the most simple level of chromatin. The DNA double helix then gets wrapped around the nucleosome, which has a core of 8 histone proteins. DNA wraps around the nucleosome approximately 1.65 times. The nucleosome then becomes associated with an additional histone protein, histone H1. The nucleosome plus the histone H1 forms a structure that's referred to as the chromatosome. Chromatosomes will then coil up further to form a 30 nanometer chromatin fiber. These 30 nanometer chromatin fibers then form loops with an average length of 300 nanometers, and these loops are then further compacted. These compressed fibers are 250 nanometers in width. Further compaction of this fiber forms the chromatid, which forms the chromosome at the end. So as you'd imagine, with this level or degree of coiling, at the chromosome level, when it's most tightly coiled up, very little gene expression can occur. Because in order for a gene to be expressed, it's the double helix or the double helical sequence that needs to be read by transcription machinery. And therefore, understanding that DNA can be compacted and coiled to this level in order to fit into a cell will help us understand how which regions are uncoiled will regulate expression of genes. So by now we would have learned that chromatin can be either heterochromatic or euchromatic, and these are very broad terms that refer to supercoiling of DNA. Heterochromatic regions of DNA are highly repressed, and that's because they are very tightly coiled. When DNA is very compacted or coiled, that DNA cannot be expressed to form mRNA or protein, as transcription machinery cannot access that region of DNA. Euchromatic regions of the genome are slightly less coiled. The term euchromatic DNA will refer to DNA that is less compacted. However, even within the realms of euchromatic DNA, we have varying degrees of compaction and decompaction. Therefore, understanding how and when the genome is unpacked is important in terms of understanding gene regulation. We also know that genes occupy distinct positions on chromosomes. So you get chromosomes, and certain regions of that chromosome may be gene dense, and other regions of the chromosome may be gene poor. We also know that chromosomes occupy distinct positions in the nucleus, and these distinct positions are referred to as chromosome territories. The positions of chromosome territories can vary between cells within a population or during different stages of the cell cycle. Chromosomal DNA can cluster into topology-associated domains, and these domains have stochastic or variable positions. TADs contain chromatin with varying degrees of compaction. This diagram shows how chromosome territories and chromatin domains can form within a nucleus. So if we start at the beginning with double-stranded DNA, which is represented by this red string, the double-stranded DNA is first wrapped around the histone proteins to form the nucleosomes. Certain portions of DNA can unwind or become separated from the nucleosomes, and these unwound segments of DNA can form chromatin loops or clusters. These loops can be variable in size, and this allows for different regions of DNA to associate with each other at different points in time. These loops and clusters can occupy specific positions within the chromatin domain. So we refer to these different localized positions as compartments. So each of these domains within a chromosome, so this part of the diagram represents a single chromosome, and each of these domains can occupy a different position. And these domains can cluster at variable positions at different points in time, which means that they are dynamic. So these TAD domains or TADs move around within the chromosome. So you have clustered regions of genes, and these different regions of the chromosome, which are represented by the different colors, can occupy different positions within the domain of the chromosome at different points in time during the cell cycle. However, chromosomes still occupy distinct positions within the nucleus. So chromosomes have territories within the nucleus. This part of the diagram here, the circle represents a nucleus, and each different color represents a specific chromosome. And these chromosomes will occupy a distinct position in the nucleus, meaning that you won't have large amounts of DNA from one chromosome occupying the territory of another chromosome completely. However, these chromosomes can shift around within the nucleus. So if we look at the yellow chromosome over here, this chromosome can occupy different positions within the nucleus at different points in time. However, it will still occupy the same region that it was in, meaning that it will still have a territory of its own, even though this chromosome can shift around in the nucleus. Going back to this part of the diagram showing the chromatin domains, we have these different territories, or we have these different TADs within a chromosome that can cluster at different positions at different points in time. And so what we know from this diagram is that the positions of different regions of DNA within a chromosome can move around, and the chromosomes themselves can shift their position within the nucleus. And considering that these different compartments can shuttle around or move around within the nucleus, tells us that movement of these chromosomes or chromatin domains within the nucleus may be linked to gene regulation. If we consider these regions of overlap here between the chromosome territories at the ends. So let's look at the green and the blue regions. There's a partial region of overlap between two different chromosomes. And if certain TADs were to cluster at the periphery of these chromatin domains, this will allow for interaction between those two chromosomes, meaning that if a transcription factor is expressed somewhere at this location, it could have access to two different chromosomes or genes on two different chromosomes. Therefore, the movement of DNA within a chromosome compartment, or domains or regions of chromosomes within a chromosome compartment, as well as the dynamic movement of chromosomes within the nucleus, all serve to functionally regulate gene expression and play a role in transcriptional regulation. So if you would like more information on this concept of TAD domains, I would recommend that you follow this reference. You can search for the article, Molecular Basis and Biological Function of Variability in Spatial Genome Organization. This article has not been included in your resources as it will not be tested. However, this diagram will be included in your course syllabus and material. And that completes this lecture. Thank you.